Dr. Francis actually is going to speak about the wealth of such resource in Guyana when China and USA and the other countries who are infected with coronavirus don't even want to hear about the wealth of microbes. But never mind, at this juncture, we're basically going to hear quite a lot about microbes, of course, including viruses, bacteria, fungi, and other kind of microscopic organisms like protoplasms and those other types. Before we proceed, I just like to basically housekeeping rules at the moment. You have the washroom for those of you who desire washroom. We also would like to say that this session would last for roughly an hour. Dr. Francis will present, and then afterwards we'll have our discussion. But before Dr. Francis presents, we would like first and foremost to invite to give the welcoming remarks our own Professor Paloma Mohammed, Deputy Vice Chancellor of PACE and Chair of the UG's Transitional Management Committee, to give welcoming remarks to all of us this afternoon. All right, Professor Paloma Mohammed.
Good afternoon, ladies and gentlemen, and it's wonderful to see so many young people here, and I'd like to pay homage to those also who are young at heart among us and younger. This afternoon I have the distinct pleasure of introducing one of our university and national treasures, Professor Pat Francis. Many people would not know that last year, Professor Pat Francis, who is a PhD in micro, whatever this thing is here. I am a social scientist, I'm not going to try and say it. But she became the University of Guyana's first ever professor of practice. What is a professor of practice? The university has decided that people who have spent their lives working, creating excellent value, doing research, supporting community work, supporting innovation, and basically creating a place and a cradle for knowledge in the country should be awarded and accorded the highest and most prestigious positions in the university. And Professor Patsy Francis was chosen to be the first of such. There will be 5 this year, and she's the first. I believe that you should give her a rousing round of applause. Many people know Patsy, and she was quoting about this photograph that we put from, she says, her Ruction days. This was from, many people know her from her work in the union and her great advocacy and her big heart for human beings and caring for others. Many people do not know that she is, in fact, a very, very, very well-grounded and distinguished academic. And so this afternoon, this is what we're going to hear from her outside of the Ructionness, but into her academia. As the professor of practice for innovation and entrepreneurship and research in a new institute called UGIRE, which is what she's going to head, she will bring to the market and bring to publication, amongst other things, many of the research projects of other University of Guyana academics that have been lying around for over 20 years without attention and without, yes. And this is not something that the university thought of. This is something that Dr. Francis, Professor Francis, I have to get used to calling her that, Professor Francis has been working on quite by herself, relentlessly, for 20 years herself. And so finally, her ideas, her passion, and her commitment have come together with the right conditions at the university to help us to create this institute and to give her a position that she should have had, I believe, maybe 10 or 15 years ago. So ladies and gentlemen, without saying much further, I would like to say also that every Wednesday, from now until the 27th, I believe, of February, this venue from 6 to 7 p.m. will be the home of several professorial lectures. She will be the first. There are lecturers who are working in social sciences, in politics, in agriculture, and so on. And so I'd like to invite you to come along. It's free, of course, and we have refreshments. We can provide all kinds of other good entertainments for you otherwise. In this year, when the country is 50 years old as a republic and the university is 57 years old, you will see on our flyers and our memorabilia and our advertisements the logo 5057. That's what it means, 50 years as a republic for Guyana and 57 years for UG. There are a number of other events that we have. Younger people might be interested, and not so young at heart might be interested as well. There will be a documentary series that have been created of films and documentaries by our students in the Center for Communication Studies. This is happening this Saturday and Berbice, next week Saturday in Linden, and then finally the final Saturday here in Georgetown. All day you can come in and you can see documentary after documentary about Guyana and Guyanese life. We will also stream for one day only at some point in the future before the 29th of February. There will also be a diaspora conference and also a conference on, I think, race that the Dr. Thomas Singh and his family, his department are doing. So a number of things going on. Please feel free to check our website and to contact and ask anybody around from the university as to the dates and to what you can do. Once again, it gives me pleasure to see Dr. Francis stand in her rightful place as professor of practice and to do her academic work and to commend this lecture to you. It will also be online, and she's probably going to do it again, probably in Linden or Berbice at some point in the future. The next person who is going to come to the podium also is as equally distinguished and dear to the university and the country as Dr. Francis herself, and they're old friends. He happens to be our Pro-Chancellor, Major General retired Joe Singh, who has a wealth of knowledge, but also has a special place today because he and Professor Francis go a long, long way back. And so he said he was delighted to come and introduce her. He's not only going to introduce her, but he's going to say a few words as the sitting chair in the absence of the Chancellor of the University of Guyana. So please welcome with a warm round of applause, Major General.
Good evening, everyone. Good evening. The mic is gone? Is it? Oh. Is it better now? Thank you. Good evening, everyone. Distinguished audience and those who have made the effort to be here this afternoon, I'd like to join with both Dr. Elroy Charles and Professor Paloma Mohammed in welcoming you here to this first of the series of professorial lectures. Particularly for young people who are here, Professor Mohammed spoke about the work that Professor Dr. Pat Francis has been doing for over 4 decades. She started, like many of you students here, at the Guyana School of Agriculture, went on to the University of Guyana, got her degree, first degree, and then went on to the University of Tennessee, where she did a master's degree in animal nutrition, and then went on to do a doctor of philosophy in animal nutrition and microbiology. And I think it's the latter aspect that we'll be hearing a lot from her this afternoon. She is one of the gems of Guyana. There are others who, many of you young people, if you don't know, how many of you here have read Winslow Davidson's book, 50 Years in Agriculture? What? Winslow Davidson was the principal of the Guyana School of Agriculture for nearly 4 decades, and he wrote a book, 50 Years of Agriculture in Guyana. that should be compulsory reading. He passed away this year, a gem of a person, wealth of knowledge. And I believe that, I'm not trying to get at anyone here, but this is the problem. We are not recognizing persons who are in Guyana who have pioneered work and who have gone that way before, who have set the pathway for us. I want to pay tribute to Professor Mohammed and the department that she heads, Space, and her team, for seeing the merits of having such a program. For too often, we feel the university is them and us, and we need to break down those barriers. This is an attempt here to influence thought leadership, to ensure that what we do in academia has an application in the real world. And you students who are here are what I consider to be the persons who can make this happen. You're not here just to get a diploma and then go on to the university. You're here because you should be seeing yourselves as persons who are going to make that Guyana transition from what it is now to the vision that we have of a Guyana of the future. A Guyana which has oil and gas, yes, but which is not going to be totally dependent on oil and gas, which will be diversifying its economy to ensure food security, to ensure value-added chain of products, such as she will speak on today, and that we will ensure that our infrastructure, our education, our health, and all the other things are equipped for the world of the future. So today we are very privileged to hear from Dr. Francis on a very important subject, and I say so from my privileged position in that I sit on a number of other boards of agencies like IPED. Some of the reasons why we have not been able to move from theory into practice is that either word doesn't get out to the people who are prepared to experiment, who are prepared to take risks, so that we develop things, whether it's at the university or at the technical institutions, and it doesn't really see the light of day. It makes you eligible for a diploma or a degree or a certificate, but it stays there. This is an attempt to remove those barriers and to put this in the public domain so that persons can now run with this from an idea into reality. We have another campus, Obtain, in Region 6. And last year, I attended 2 workshops run by Professor Subramaniam Gomatiyadian. And one of it had to do with mushrooms, and the other had to do with value-added products from milk. And it was amazing to see the interest that those workshops generated among members of the community. So much so that a few of them are actually running their own mushroom houses and are experimenting with different milk products, yogurt, etc. What I would like to see is that Guyanese, some of them unemployed, some of them who are looking for a niche area in which to develop micro-enterprises so that they can make a living and so that they can be part of that value chain. So this, I don't want to steal Dr. Francis's thunder, but I will ask you to listen, both the mature persons who are here and the students who are here, to what is possible. Dr. Mohammed, Professor Mohammed mentioned about the 50th anniversary of the Republic and the 57th anniversary this year of the university. I think the theme which has been reflected in these professorial lectures is vision. The I is for, what is it? VI and evolution. So that we have a vision, we want to share this with you, and we want to see how it will evolve. Are we sowing the seed on fertile ground so that we can move and take it forward? So I have great pleasure in inviting a very distinguished lady, daughter of the soil, someone who I've known for over 4 decades and for whom I have the utmost respect. Listen to her attentively, but don't just listen and leave here. Listen and be motivated to take action, either in your own way as an institution, but also when you graduate in influencing that thought leadership and taking people out of poverty and letting them see new avenues, new pathways to development. Thank you.
Yeah, we'd like to certainly thank our Pro-Chancellor, Major General Retired Joe Singh, for his kind remarks of Dr. Francis. Even I myself, a protege of Dr. Francis, coming through from the time of university, I can remember Dr. Francis said to me, he said, Elroy, you know, we should really form a agriculture club. Right, and I would like to be part of that club. And so she urged us and organized us, and then we formed that club. Incidentally or accidentally, I became its first president, the first president of the Agriculture Club. I think Dr. Francis is like, I can't do without her, or the reverse, she can't do without me. Which one, I don't know. But subsequently, when I became a lecturer at the University of Guyana, she came back to me, but this time she was saying, you know, we should really move agriculture. We should really do something about agriculture in the country. And so she pushed the then Vice Chancellor to have a plot of land for us to develop our own farm, because she said, look, once you have this farm, you can basically see what's happening. Students can come, because we lack finance at that time to go and visit other farms, so we can do everything there. And she pushed hard for this to happen. And you know what she did? She certainly co-opted me into her arrangement, and I became actually the first organizer for our research center plot at that time. And so we moved that research center, and you should come to see what it is like today. Afterwards, I can remember, well, roles reversed. She was, no, I forget this one. I can remember when we had, you know, there were the funding being given from Commonwealth, and the university was saying, but we don't have persons to apply for funding. I can remember Dr. Francis pulled me aside and said, Elroy, why haven't you applied? I said, man, Dr. Francis, I'm not sure whether I want to be with university that long, right? And so she said, well, I don't want that excuse from you. I want to see an application immediately. Right? And so I sent an application without the intention of, you know, getting the scholarship or anything of that sort. Because she asked for it, I just sent it. Right? And I sent in the application, but lo and behold, I got the scholarship. Right? And so I went to UK. When I came back from UK, right, I was the dean at that time within the faculty. And so she was saying, you know, for a long time, we needed to have this master study program. You know, for a long time we tried to have that master study program. I think you and I can make it work. Right? And I said, okay, Dr. Francis, since you want to push that master study program, here's what's going to happen. You make it happen, and I'm going to push for it to occur. And so Dr. Francis, you know, she sat there, she put the document together. I took the document, and we ran with it. And then we started the master study program in agrotechnology and business. And in that program, what happened is that we have food science and an area which is very much needed within agriculture. And I'm quite certain this is the area that she will be speaking on, biotechnology, and it's essential and important. So I welcome Dr. Francis to the podium to talk about microbes.
Good afternoon, everyone. All protocol observed. What Elroy didn't tell you is while he was in England, I wrote him. And I said, Elroy, get yourself back here as quickly as possible. Don't go and stay in the people place. You need to get back here. And I am telling all young people that you the country cannot develop itself. We have to develop this country. It doesn't matter how much money we get and what aid we get. If you don't fix up your own house, then you might have to go over by your neighbor. And that's what we've been doing, and so we got to stop doing that. And our best brains have got to be here and develop our own country. All right, this afternoon, it is my great pleasure to talk about microbes. The first time I heard about microbes was at the Guyana School of Agriculture. And then at UG, I really didn't know what it was all about, but I recall that Dr. Paris, who was at ECA many years ago, I think it must have been 94 and 92 or some year like that, she said to me, could you come and give a lecture to the Veterinary Association? And I was thinking, well, what do these vets want to hear about, you know? I'm not a cut you and fix you up person. Anyway, I found a magazine with some information on enzymes. Enzymes used in the feed industry, and I decided that I would talk about that. Later, when I got a scholarship to do the PhD and I arrived at the University of Tennessee, and they asked me, well, what do you want to do? And I told them I'm working with enzymes. And they said to me, well, you could work with whatever you want. This is the nutrition part of the thing, but you must have some microorganisms in it and food safety. You must have it there. And so I had to marry the nutrition with the microorganisms. A nice thing that happens at UG, when I got back, the agricultural microbiology program was given to me to teach. And every year at UG, you have to improve on your teaching methodologies and so on. I don't know if you guys know this, but it is one of the things that get you promoted in part or overlooked. I don't know. But in order to bring life to the subject matter, I started to do projects. And over time, it has developed into quite a mass of information. Now, I must tell you also that there was a professor here from India. She's now working for a fancy company in the United States. And she helped me. Her name is Lalita Kumari Janarthanam. I always speak her name because it was a mushroom project. I knew nothing about mushrooms, and a student wanted to do it and was getting trouble, experiencing trouble. I think Elroy was her supervisor, or it was Dr. Velosa. Must be Dr. Velosa was her supervisor, but she was having a lot of trouble, and she wanted to give up and do something with bora. And I said, but how could you move from mushrooms to bora? It can't work. You know, you have to do it. You see, you chose it. You have to do it. And I started looking around the Caribbean and other places to see, and then I started to look through every person's CV on staff, because I was dean then, and I saw Professor Kumari CV talking about mushroom. I called her, and she said, yes, I know all about it. As a matter of fact, I have the culture and everything at Berbice. She is, or Dr. Gomati is her, he's her student. So he's now at the Berbice campus. He's director of Berbice campus. But anyway, from that time, what we did is bundled everybody up. The student, Dylan Husbands, who is doing her PhD now in fungal systematics, you know, all these things. And 5 other students have passed through the faculty doing mushrooms. And I'll tell you more about that, but we haven't made the strides that we should have made with getting the mushrooms into the market. So that is one of the things. So it is Professor Kumari and other people like her, Dr. Gomati too, they talk about microbes. Then there's some people who came into the country and they set up a facility where they're importing microbes of different types into the country. They're doing it then and they're still doing it now. And different people have come into the country and are taking our microbes out of Guyana and elaborating them in countries, in other countries. I can tell you that I don't have a quarrel with it because, I mean, it's our resource and we really need to be doing something about it. So this afternoon, I want to talk a little bit about what we've been doing recently. And the students from GSA, I'm very glad so many of them came. Thank Dr. Allen for mobilizing them to come, because I have been putting them through the paces of doing microbes, of growing microbes that are important to agriculture. I don't know if they realize what good work they've done, but other than the grade, it's more than the grade. It is that you have done work, and now I hope that you will take what you have done at GSA a little further, because this is the way that we move from strength to strength. So, what is this all about? Now, this is a dairy farm. This is a dairy farm that I had a lot to do with. It's a dairy farm in Barbados. I have a lot to do with these dairy farms in Guyana too. But I was very impressed with this particular farm. This is a part of agriculture. Next slide. This is agriculture. Milk processing. You see we have milk in the bottles there. That is, what kind of milk? It's pasteurized milk. This is low temperature, long time. And I don't have any high temperature, short time there, which would be in the boxes. You guys know all about that. We have cheese there. We have yogurt. This is agriculture. We have aquaponics. We have aquaponics at UG. We have aquaponics at GSA. Next slide. We have raised beds at GSA. We have raised beds at UG. Next slide. We have modern agricultural greenhouses all over Guyana. Next slide. Next slide. Okay, so the type of microorganisms that we're looking at are bacteria, and they're very good bacteria, and this actinomycetes, which is somewhere between bacteria and fungi, you know, they're Douglas. We have fungi. Fungi is such a wide, very big family, fungi. Got to keep them out of your hair, got to keep them out of your toenails, but they're good ones, as you guys have found out. Then we have algae. Algae is a master producer, some of them, of nitrogen. Then we have protozoans. We have good protozoans, and we have bad ones. Now, our cattle have good protozoans in their rumen, and this is what helps to give us the milk that we drink. And these protozoans, algae, fungi, actinomycetes, and all of the others, they all come from the soil. They're all in our soil. Okay, now for this academic year, 2019-2020, these are some of the projects that were done. Some were done at GSA, and some were done by the students at UG. I am their guide. So carambola wine, I can tell you these things taste real good. Cashew wine, sweet potato beer. Man, this thing is for real. The sweet potato beer along with Banks DIH, you were able to produce that, very nice. Soft cheeses and hard cheeses, yogurt, and compost. Those were some of the projects that were produced in this academic year. Then also, we decided to go even deeper, and as you would see here, the projects that we did, growing and identification of Azolla anabina. The Azolla is a fern, the anabina is the algae. They grow in a symbiotic relationship, and they grow in ponds, in ponds all over Guyana. And I'll tell you a little bit more about them. Then they have the rhizobia. They did preparation and application of rhizobia species. These are nitrogen fixers. And the rhizobia is found right here, and the students were able to select them from the soil and grow them up in the lab and multiply them and then apply them to crops and then evaluate how the crops performed. Then we have the trichoderma. Now, trichoderma is a master. The rhizobia is a bacteria, the trichoderma is a fungus. Now, the trichoderma, this organism has so many applications, as you would see. Then we had one student did, a group of them did a construction of a biogas plant. I wanted to show them that the microorganisms are already there in the manure that they're feeding the plant with, so that the gas that is produced is actually produced from microorganisms that are already present in the manure. So they actually constructed it and a microplant and did that. Then they did, you have to talk a little bit about bad organisms. Like this one is a big problem. Clostridia perfringens, a big problem around the world. Causes a lot of problems, and they are resident in the chicken gut, and so some students looked at that to see if we had them. Okay, then the biostimulant using lactobacilli. Now, lactobacilli, as a former student of UG said to me, he said, "Miss, this is a master organism." And I'm sure that most of you know that the lactic acid bacteria, that they do very good work everywhere. In your gastrointestinal tract, everybody eats yogurt. Make sure you buy the one with the active, that is marked active. If it's not active, it's just the flavor you're getting. So make sure that you get the one marked active. Okay, then the biostimulant using the Saccharomyces cerevisiae. Now the Saccharomyces cerevisiae, which is your yeast that you make your bread with, this Saccharomyces cerevisiae is also good for making the wines, but there are many different species, and they do all sorts of jobs, as you would see. Then we did some evaluation of food safety, and the students made their they multiplied a starter culture in order to know that they don't have to keep buying the culture from a factory abroad. And as time goes on, we should be able to freeze dry these, the culture so that we could have them in the packet and use them. But for now, we have to extract them from wherever we can. Even from the calf's stomach, we can extract them from there. But just buy a packet from somewhere or buy it in a yogurt. Next slide. Okay, all of these organisms are beneath our feet in the ground. We have very rich soils in Guyana, all over Guyana, even our sandy loams on the Essequibo coast, because some of these projects were done over there. And I was surprised to find that even there, many of these organisms are already present in the soil. So it's not only on the clays, but even in some of the sands, we have these organisms. Next slide. Okay, now these microorganisms in the soil, they have many different things that they do. And I don't want to bore you with the lecture because I know most of you just want to hear, what can I do with a microbe when I leave this lecture? So we're not going to go into all of this stuff. And at any rate, you'll probably get it on, all of these young people know all about it already. And the older people who may not know about it, they can always read it up on the internet or on the flyer that we will give them. Okay, now the algae. Now this Azolla anabina, it's free-floating in our drains, and thanks to Dr. Austin, he is an animal scientist with GLDA. He took the students out because he knew exactly where that organism was and helped them to gather it up. Now, in other countries, this Azolla anabina is used for many different applications, for fertilizing rice paddy fields, fertilizing different crops, and even use the anabina is even used for food in some cases. And it multiplies very quickly, and it really does not need very much nutrition. It is photosynthetic, so it means that it is making its own nitrogen, which is great. And one thing I must say that IPED has a handout, a little booklet, you can find it online, where they're talking about using pondweed. Now, pondweed is also good, and very useful for feeds. But this anabina, it's a marvelous resource. And what the students were able to do, they were able to grow it up using 2 different methodologies. And they also, they harvested it, and then they packaged it. So right now, if you go up to the Guyana School of Agriculture, you'll find it in a packet. They're sitting in a special place. So I know you're going to say, when is it going to be in the shop? I got to depend on the university and the government and other people to help us to get it there, and on you, young people. Okay, so many things it can do. Next slide. I've told you all of this already. Next slide. Okay, I like pictures because they speak 1,000 words. And there you see in the left-hand corner, I don't know if the student is sitting there, or the group, but they were there collecting it. They were in some kind of minivan that probably was arranged by Dr. Allen. And they went out there, and you could see they collected it from the field. And then they came back in the lab, and they grew it up. And they did all the lab work, and then eventually they packaged it. Now, where do we go from there? Knowing all the things that Azolla anabina can do. Next slide. Okay, now for the fungi, there are so many, and I don't have enough time to tell you about all of them, so I'm going to speed through a little bit. These other speakers took up some of my time, So, so the trichoderma, the rhizopus. Now, rhizopus is extremely common. Rhizopus is what happens when you leave your bread outside or you forget your cheese somewhere. So rhizopus is all over the place. Rhizopus is a master decomposer. And we have rhizopus. I mean, if you just slip a rhizopus, it will grow on you. So what are we going to do about rhizopus? Trichoderma, we got to do something about that. The Saccharomyces, we can either collect it from the soil ourselves, or we can just go ahead and buy in the mother culture, because we don't want to have to start from scratch with everything. Buy the mother culture, and then subculture, and then, do what we want to do. Make our stimulants and so from that. So, mycorrhizae, I know that Nari is doing some work with mycorrhizae. I think UG has also done some, other than myself, have done some work with mycorrhizae. Not like a student in my class did mycorrhizae some years ago, but I think in recent times more mycorrhizae work is going on. Mycorrhizae basically it just extends the roots of the plant everywhere, so that if you see those big trees, that even in the drought, they don't die. So it is something you can inoculate the soil with. And you can put it, I think Nari is putting it into a mix that they're making right now. But these things need to jump out of the lab and jump into the shop. All right, mushrooms, we have lots of mushroom, oyster, paddy straw, many, many different kinds of mushrooms. And you want to hear a secret? We have mushrooms that no one else has in our interior. One of our students, he and his husbands and so on, had arranged for them to go up there into Waramadong, and they were taken with by a guide and shown all these edible mushrooms. Now, our indigenous people know which mushrooms are edible, so don't just go eating the jumpy umbrella, you know what I mean? So, what are we doing about these mushrooms that we have that are edible? The world is waiting on the fungi that we have, and they're very full of protein. I remember one year, a former Prime Minister, Heinz, guy's probably too young, probably don't know him. We had a big expo in the Faculty of Agriculture. The Faculty of Agriculture is an amazing place. When we get it right, we got it right. And we even had a curry mushroom, which the Prime Minister tasted, and so on, and still, it's not in the shop. All right, trichoderma is a very wonderful fungus. One of the good things about trichoderma is that it not only helps the plant to grow well, but it stimulates the plant to grow, keep off other organisms from attacking the plants, but it also protects seeds and during the germination process. And so this organism, the first person I told you I saw produce this organism was Professor Lalita Kumari Janarthanam, and she presented a bottle of the liquid, the insecticide, which was supposed to help us with paddy bug. You know that paddy bug is a problem until now. And nobody took it up. We still have the recipe. Once she gave that 1 gallon worth of trichoderma liquid to the minister, maybe the reason why it went nowhere, he was killed. Saturday Oso, I call his name. He was my friend. He was a good minister. And I suppose he died, and so nothing happened. But that doesn't mean we're still alive. And you have been working on trichoderma. Right there at GSA's farm, you went out and you collected soil from different areas, and you found that there were many different fungi growing together. And from it, you were able to isolate. Trichoderma has many different species, and certainly you were able to isolate one species. And trichoderma powder, which is the next step, is used all over many developing countries, including emerging economies such as India. If you go online, you will see trichoderma powder is up for sale. You can buy it in the shop. And that is what I'd like to see. That's where we should go. All right, next slide. Okay, importance, keep going. Okay, these are the students. I don't know if they recognize themselves. They're extracting trichoderma. There are two groups. You never knew that you would turn up in lights, eh? All right. So there they are doing the work. Next slide. Isolating. Next slide. This is what they were supposed to see. Some of them didn't see this, but one group did see it. So we knew we had the right thing. Next slide. And this is the final product. Now, they did not produce the final product, but I know the final product can be produced because it's been produced right here before, as I was telling you. So we need to get it out, either the powder, in powder form or in liquid form, so that we can give it to farmers or sell it to farmers. This giving away thing is not good. I hate free. Next slide. Okay. Now, mushrooms, as I was telling you, since about 1994 or some years like that, Professor Kumari, she helped us with this, and we have had at least 5 or 6 students work with this mushroom project. And this mushroom project is quite involved. That is why I didn't give it to the GSA students to do, because it needed a clean space, a sterile space, which I didn't have at GSA. Okay, so this here, you get the mother culture, and you can buy the mother culture, and you have to then grow it up like this. It's very, you have to be very careful. Because as I told you, if you miss, rhizopus will grow on you. So rhizopus is right around us right now. So it's looking, you just put down something, it's there. So you have to have a clean space to do this work. Next slide. And then you have to go to the mass multiplication, and then to the spawn culture, and to the mushroom house, which, as you can see, we had the mushroom house. I think it was taken down only recently at UG, where we have the next slide, where those beautiful mushrooms were grown. Hundreds of bags. Over many, many students grew that and others. We even had seminars for people in the field. But as I was explaining to Major General Joe Singh, the problem with mushrooms is that we cannot expect a person, an entrepreneur, every entrepreneur to set up a clean space. It's just not going to happen. So there must be a clean space somewhere. And I think the university is poised to have a clean space and to have a biotechnology space where all of these things can go on. And then we just give the spawn, which is in the bottle, to the consumer, to the entrepreneur. Then they take it and inoculate their beds. Now, these beds are made out of different organic waste, such as coconut fiber, straw, different things we tried. And so this is how mushrooms have to go on. Now, some mushrooms need a cold period, some degree of cold. And we do have parts of Guyana in the mountainous areas that are pretty cold. And I would say, let the mushrooms grow where they will. In the cool places, we grow the button mushroom. Everybody in here, including myself, like to eat pizza. I've never bought any of the local pizza because I got spoiled by my Italian pizza making, my friends, I don't think anybody locally, but I'm a bit biased. But on those pizza, they have the mushroom on there and you like it. And all of those mushrooms, all mushrooms that come into Guyana are now imported and have been for some time. Now, this mushroom used to be in the Nigel supermarket and some other supermarkets because Miss Husbands, she had packaged them and so on and so forth. But unfortunately and unfortunately, the right carrot was swung in front of her. Come and get your PhD. So what could she do? She had to drop this and go behind that. But she'll be back. She'll be back here in another year or so. And I'm hoping that by the time she gets back with her PhD in this very area of fungal systematics, now she knows a lot more about fungi than when she left, that this business could get going again. Young people also, and older people, a lot of people have been asking, what are we doing about the mushroom business, And as I tell them, I was telling Major General, that really what we need is a clean space. a biotechnology space where all of this trichoderma and all of this azola and all of these things could be happening. Teams of scientists, whether they be geneticists, whether they be productionists, whatever. There are biotechnologists, people who are extracting enzymes, big vats, fermenting and producing from our soils all of these products from our own microorganisms that are right beneath our feet. Next slide. Okay, so I've told you about these bacteria, lactobacillus. We made a, next slide. These are students producing the lactic acid bacteria, which is a combination of Lactobacillus acidophilus and Bifidobacter and different ones. And I love these organisms, can't you tell? And, and these, you like them too. You just like to eat them. You don't want nothing to go fixing and flipping with them. You just want to get them in a packet. Now, these students were producing that lactic acid bacteria there. They had their concoction and so on. And in this, next slide. In this, you would see me standing there. That's in a field in Barbados there, myself and the farmer. That's the kind of entrepreneur that I want here. You know, look into it, trying to get it. And there on the tractor, on the top, you see a small dispenser. It's up there. Could you see it? Yes. In that, the lactic acid bacteria is in there. That concoction that you made, it's up in there. And as the grass is cut, it is sprayed onto it as it is being cut, and it is being loaded into a tractor. Now, what that does is facilitate the lowering of the pH and the keeping off of all of these other organisms that we don't want in that mass. And so it helps to cure the mass, which you know is silage. We don't have any of this in Guyana. But it's not that we don't know how to do it, you know. Why we don't do it, I'm not sure. But that is the end product. And then you can, during the dry season or in the wet season, you just take that out and you feed your animals. It's done all over the world. Next slide. All right, the rhizobia, these students, with the help of NARI, they were able to go into the field, collect the organism from the soil, come back into the lab, prepare all the doodads. Next slide. And then they produce the final product, which is sitting there at GSA. Next slide. That is what the rhizobia looks. It fixes nitrogen. It lives in a symbiotic relationship with the plant root, and it is found naturally in our soils. And we've been putting it there for such a long time that to recover it from the soil is not that difficult. Next slide. Okay, we got a lot of waste. The coconut waste is tremendous. The household waste is more than 51%, the big blue part. More than 51% of the waste that we have going to the landfill. Next slide. And our fertilizer imports are great. So what are we going to do about all of these microorganisms? Let me see if I got any more slides. The way forward. Okay, so the way forward, I believe, is to continue doing what we're doing now, getting young people interested and working on these microorganisms, let them get a feel of things in the laboratory, talented teams working on more exotic, more difficult things like extracting enzymes, like the ones that I worked on in my PhD. You know, that big company called FinFeeds, they sent me the enzymes on a medium, which I applied to a grain, and then this grain is full of cellulase, which chicken can't break down. So the enzyme breaks it down. So when they sent me the enzyme and I apply it at very low percentages, like 0.1% and 0.05%, 0.05%, very low percentages. And there you are. It releases the nutrients. And at the time, I thought when I came back to Guyana that I would get the feed companies to buy this thing. But why I haven't gotten them to do it, I don't know. Anyway, as I say, there's life and there's hope. We need improved biotechnology lab. We need a space where right now Dr. Charles and his other colleagues, they're struggling in a small space that they've worked very hard to get that space in collaboration with other universities. They wrote their own project, and so it was a little space that I had put together for the mushroom, and they elaborated on it. And now they're bringing in all this equipment that they were able to get through their own efforts. And really and truly, they should be encouraged by being given a much bigger space so they can do a lot of work. And many scientists can come and work. You know how they found Miss Husbands? People came to Guyana and met her right there in the faculty doing work. They could not believe that she knew all those things. And with the help of the dean at the time, the dean again now, scholarship was arranged. But when she went abroad, they were shocked to know that she knew all of these things about fungi, you see. But we use what we have, all these old microscopes, and so we have little better ones now. But the people brought the equipment here, all the universities, because we have the talent, we have the brain, we have the soil, we have the resource, but we need some assistance, some better help. I recommend very highly a space for biotechnology. Many years ago, when Professor Kumari was here, I had envisaged a biotechnology park where we would, up in Berbice, where we would have been producing all these things and so on. But that didn't happen. Anyway, where are we going to get the money from? We're going to look for the money. And Dr. Mohammed knows how to get money. You know, we depend on her to look for some more money, and we will look too. And so, hopefully, some of you, I know many students who passed through my hand have gone on to do something with microbiology, but we have to do more. And you have to keep manipulating. You might say, Miss, I get the trichoderma when I was in the lab and so on, but now that I'm graduating, what I can do? I'm going to work at this place. The people are not doing that. They don't have no need for that, and so on. Think about where you want to go and put yourself there. Your employer does not own your brain, and if they have no use for it, please use it for yourself. You should be using it for yourself anyway. And so, I think that is the end of my discourse this afternoon. Thank you. And